So our next speaker is Louise from UC Berkeley. She's in the plant and microbial biology department. She's a full professor there. She's also part of the Energy Bioscience Institute at the Berkeley lab, and she's going to talk about, I believe, studies in the model Filamentous fungus, Neurospora, and plant cell wall deconstruction.
Okay, thank you very much. I really appreciate the opportunity to tell you about our work today. I am going to be telling you about our understanding of plant cell wall degradation using the model filamentous fungus Neurospora crassa. Now, Neurospora crassa has been used in the laboratory since the 1920s, primarily as a model organism for genetics, biochemistry, and biochemical genetics. And it's been funded primarily by NSF and NIH, work on this organism. But in the wild, this is where you'd find Neurospora. It's always associated with dead plant material, primarily grasses in this case. So we have a lot of isolates from burnt grasses and burnt sugarcane. But although cellulases had been recovered from Neurospora in the 1970s, there was virtually no work on this organism on plant cell wall degradation since that time. And so the EBI, which is a joint venture, which I'll talk about in a minute, came to Berkeley in 2007, and so John Taylor and I and Tom Bruns decided to submit a grant, and I was interested in understanding the plant cell wall degradation program in Neurospora crassa, and I was a little embarrassed about this grant because I thought everything was understood already, and I would get totally knocked down by submitting this grant, and I've been pleasantly surprised that there's actually a lot that's not well understood, not only about the structure of the plant cell wall, but how filamentous fungi are able to degrade plant cell walls. And so I'm going to be telling you about that story today. So first, to tell you a little bit about EBI. Okay. So the EBI is a joint venture between the University of California at Berkeley, the Lawrence Berkeley National Lab, University of Illinois at Urbana-Champaign, and British Petroleum. And it covers a really wide variety of approaches to developing biofuels as well as recovery of fossil fuels. And our group is in the biomass polymerization part of EBI. So it's not, oh, I know. Let's see if that works now. Okay, so there are a lot of processes associated with turning lignocellulosic material into fuels. And many of these steps have not been worked out, and many of these steps, no one really knows what the final version will be. But the first thing that has to happen is you have to take this dry plant biomass and chop it up into little pieces. That takes energy. Then you then have to have a pretreatment. There's a variety of pretreatments that are under investigation. No one really knows what the pretreatment will be. But the purpose of the pretreatment is really to remove the lignin from the cellulose and hemicellulose in the plant cell wall. Currently, that hemicellulose and cellulose is then enzymatically cleaved into C5 and C6 sugars. And once you get those sugars, it really opens up all sorts of opportunities because you can use a variety of organisms that are able to ferment sugars into a variety of fuels, including ethanol, butanol, and other fuel-like derivatives. So this part, this hydrolysis of cellulose and hemicellulose, is primarily done by filamentous fungi. So in nature, the primary degraders of plant cell wall material are filamentous fungi. And this is Trichoderma, the currently industrially used organism for cellulases and hemicellulases. But many filamentous fungi degrade plant cell wall material. So here's a basidiomycete, and here's my favorite organism, Neurospora crassa, growing on a tree. Now the reason, if you take wild-type Trichoderma and wild-type Neurospora, and you grow them on plant cell wall material, they have similar enzymatic activities. They secrete similar amounts of enzymes. But the reason why Trichoderma is used industrially is that it was isolated from during World War II, and it was mutagenized heavily. So instead of secreting milligrams per liter of cellulases, as Neurospora does and wild-type Trichoderma does, the industrial strains of Trichoderma secrete hundreds of grams per liter of these enzymes. And so that makes them the preferred source for cellulolytic enzymes for biomass conversion. And so one of the projects that we have in the lab that I'm not going to talk about today is mapping the trajectory to the hypersecretion state using Neurospora as a model. So why use Neurospora? Well, Neurospora has many tools that allow it to interrogate functional analysis of genes encoded in the genome. The genome was the first filamentous fungal genome sequenced. It was published in Nature in 2002. And this is the hyphal network. These are the asexual spores, and this is a sexual reproductive system. So it's very easy to do genetics in the laboratory. It has a long history of genetic and biochemical methods. And importantly for us, it has a near full genome deletion set. So there are 10,000 genes predicted in the genome. We have over 12,000 strains where each of those genes have been deleted. And even essential genes can be maintained in a heterokaryon in Neurospora that you can skew to look at biochemical function of essential proteins. There are mature cell biological tools. There's a large and active research community. And again, in nature, its ecological niche is degradation of plant cell wall material. So when we think about a model of how filamentous fungi break down cell wall material, if you have cellulose or lignocellulose in your environment, that's an insoluble substrate. And so somehow the fungus has to sense that it's on that insoluble substrate to be able to degrade it. So the organism, when it's under starvation conditions, will secrete a small number of lignocellulitic enzymes, cellulases and hemicellulases, and those enzymes produce small molecules that are then taken into the cell that signal Neurospora and other filamentous fungi that it's on lignocellulose material, resulting in the induction of genes encoding cellulases, hemicellulases, transporters, and other proteins involved in breakdown and utilization of this plant cell wall material, secretion of all those enzymes outside the cell that then break down the insoluble substrate. So the story that I'm going to tell you about today are about what we know about the transcription factors that are important for regulating these genes, how Neurospora senses it's on plant cell wall material, and how that might signal through these transcription factors. So the first thing we did was determine whether Neurospora, what its response was to plant cell wall material. And so we grew Neurospora on miscanthus over a 10-day time period and did transcriptional profiling using microarrays that were developed in my laboratory to look at gene expression over this time period. And what we found is very early, so 16 hours after you inoculate asexual spores onto miscanthus, that's when you get expression of predicted plant cell wall degrading enzymes, such as cellulases and hemicellulases. Now, this is just a functional category analysis of this approximately 300 genes that were induced very early and co-expressed with cellulases and hemicellulases. And as you can see, there are the expected proteins, genes encoding proteins involved in extracellular utilization of polysaccharides. But there's also a big portion of these genes that are unclassified. So these are genes encoding proteins. Sometimes we know they're proteins because we've done a lot of proteomics work, and we identify proteins that encode genes of unknown function. And so we know these are coordinately regulated, but we really don't have any idea of what their biochemical activity is. And so we're using the genomics of Neurospora, the full genome deletion set, to try to understand what these proteins are doing. And so I'm just going to give you a little side story about that process before I start talking about our work. And so 2 graduate students in the EBI who work that we coerced into working on Neurospora, Will Beeson and Chris Phillips, and Michael Marletta's and Jamie Cates' lab, were very interested in these 2 enzymes, cellobiose dehydrogenase and the GH61 families. Now, these proteins are secreted along with known cellulolytic hydrolases, but their actual biochemical function in terms of breakdown of plant cell wall material was not very clear. And so there are crystal structures even available for these proteins. And so again, Chris and Will, they utilized the Neurospora genome deletion set. And when you look at a deletion strain lacking cellobiose dehydrogenase, you find that the activity of the extracellular enzymes is less than wild type. You can then add purified CDH1, which we can express very easily using homologous expression systems in Neurospora. And when you add that back, you get a stimulation of cellulase activity. And then when you figure out in the wild type what is interacting with CDH1, you find out that it was a GH61 family. So that means that CDH and GH61 worked synergistically to stimulate cellulase activity. So after a lot of biochemistry, they determined that the GH61s actually encode polysaccharide monooxygenases, and that cellulobiose dehydrogenases oxidize cellulobiose and provide electrons to the copper-containing GH61 protein, and the GH61 protein can then oxidize or oxidatively cleave cellulose microfibrils. And so this actually began an entire new enzyme work on a novel aspect of cellulose degradation. Now, interestingly, Novozymes did a similar work, but they used trichoderma. So trichoderma has no cellulobiose dehydrogenase gene, and they used extracts from wild fungi to add to trichoderma to look for this synergy. And they identified also this GH61-CDH interaction that stimulates activity. And they published about a month before Will and Chris. But that's the way it goes. So what we were interested in, we knew that this whole transcriptional program was associated with induction of cellulases and hemicellulases, and so we were interested in what transcription factors were involved in that process. And so, as I said, there's a transcription factor deletion set for Neurospora, and so we assayed all those transcription factor deletions for ability to utilize plant cell wall material, and we identified many that were affected in their capacity to utilize plant cell wall material, ones that showed increased enzymatic activity and others, decreased enzymatic activity, and others that showed increased enzymatic activity. We characterized CRE1, which is known in Aspergillus and trichoderma to be involved in carbon catabolite repression. It also has that role in Neurospora crassa. We also characterized xylin R, which encodes a zinc binuclear cluster transcription factor, which in Neurospora is required for regulation of hemicellulase genes, similar, although in trichoderma and Aspergillus, this regulator regulates hemicellulase genes and some cellulitic genes. In Neurospora, it only regulates hemicellulase genes. But we also identified two novel regulators called cellulose degradation regulator 1 and cellulose degradation regulator 2, that were completely deficient for growth on avicell. And this just shows the phenotype. These mutants grow normally on sucrose. They grow normally on hemicellulose, but these two mutants are completely incapable of utilizing cellulose. So we needed to define what genes were induced when you expose Neurospora to cellulose. And so when you do that, initially you get a set of genes that are de-repressed. These are what we call the scouts. The enzymes are secreted by Neurospora to test, see if there's plant cell wall material in the environment, and if so, to induce the production of cellulases. So initially you get a de-repression followed by an induction. And so we did a time course using RNA-seq over this entire time course and we were specifically interested in genes that were induced only by cellulose. And from this we were able to refine the cellulose regulon to 321 genes. And so we did a similar experiment with the CDR1 and CDR2 mutants to see what part of the cellulose regulon these genes regulated. And what we found is that when you have these mutants, that the regulation of cellulase genes was totally off. So they still had the de-repression, but they did not get the massive induction that you normally see in wild type of cellulitic genes in response to cellulose. So the CDR1, CDR2 are directly responsible for regulating 204 genes out of that 320 gene cellulose regulon. 60 of those are hypothetical, and 20 of those are predicted to be secreted. So these are secreted along with all the cellulases and hemicellulases, so they must have something to do with plant cell wall degradation, but their function is unclear. CDR1 and CDR2 regulate all of the cellulases in the Neurospora genome, half of the cellulases, some proteins involved in amino acid metabolism, and it's known in trichoderma that there's this interconnection between nitrogen utilization and carbon utilization. Also some genes involved in protein synthesis and secretion, and if you think about it, all of these proteins, enzymes that are made have to go through the secretory pathway, and so it makes sense that these would be upregulated by the regulators of the cellulose regulon to accommodate that increased traffic. We're very interested in what those proteins are doing, and transporter genes. So what is the relationship to CDR1, CDR2? As I said, they both control the same regulon. And so we assessed the expression of both in both mutants. So for example, this is the expression of CDR1 in wild type. The blue is CDR2. But if you look at a CDR1 mutant, of course you don't see expression of CDR1, but you also don't see expression of CDR2. Whereas in a CDR2 mutant, expression of CDR1 is like wild type. So these data indicate that CDR1 is required for the expression of CDR2. And so this led to the following model. So if there's glucose around, none of these enzymes get made. But if there is no glucose, you have absence of glucose repression. You get a few scouts that are made. If they find plant cell wall material, they make signal molecules, which are breakdown products of the plant cell wall, that signal to CDR1. CDR1 is expressed in minimal media. So there has to be some kind of modification, we believe, to CDR1 to make it active, to turn on CDR2, and we believe both CDR1 and CDR2 are present in the promoters of the cellulases and hemicellulases because we've done some ChIP-seq analyses and have shown that that's to be the case. So that's great. So we've identified these transcription factors that are important for cellulase and hemicellulase gene expression in Neurospora, but that's Neurospora. So it turns out that these are highly conserved among Filamentous ascomycete fungi. So this is Neurospora, this is Aspergillus, and this is Trichoderma. Trichoderma for CDR1 is a bit of an outlier, but for the CDR2 tree, the gene tree follows the species tree. And so what we decided to do is knock out the genes in Aspergillus nidulans, which is about the farthest removed from Neurospora crassa as any other fungus within the Filamentous ascomycetes. And what we found is that when you delete cdrB, which is the CDR2 homolog, Aspergillus nidulans also doesn't produce any cellulases. Okay, so it's totally off for expression of these cellulases and also cellulitic activity. Whereas the function of cdrA seems to be for growth on cellobiose, and this function is also conserved in CDR1 in Neurospora crassa. And so what do we hope to do by understanding the role of these transcriptional regulators in cellulase and hemicellulase regulation? What we hope to do is be able to manipulate these transcription regulators so that we can make cellulases at will. So in other words, under minimal media conditions, under any kind of, using any kind of promoter. And so those experiments are ongoing. And so what that does, that enables you to evaluate the role of cellulases of this entire group of organisms. Okay, so I've talked about transcription factors. But how does the signal, how does Neurospora sense that it's on lignocellulosic material? And how is that signal transferred to activation of transcription factors and subsequent gene expression? So if you think about cellulose, it's a very simple molecule. You know, it's just repeating glucose units linked by beta-1,4 linkages. And when you break it down, you basically get oligosaccharides and cellobiose. And we knew that when you grew Neurospora on cellobiose, you did not get induction of cellulases. And that's true in other filamentous fungi. Now in Trichoderma, you can get induction of cellulases by a transglycosylation product of cellobiose called sulfurose, but that particular compound does not induce cellulases in any other filamentous ascomycete fungus. So it's unique to Trichoderma. So our hypothesis was the reason why cellobiose did not act as an inducer in filamentous fungi was because of glucose repression. You have these extracellular beta-glucosidases that break down cellobiose to glucose that's brought into the cell, and we know that glucose represses expression of cellulases. And so we decided to eliminate glucose repression by creating strains that were missing the extracellular beta-glucosidases. There were 2 of these that are induced when you grew Neurospora on plant cell wall material, and an intracellular beta-glucosidase. And our hypothesis was that if you got rid of glucose repression, that cellobiose would induce cellulase gene expression in Neurospora. And that's exactly what we found. Here we've done, this is qRT-PCR, but we've also done RNA-seq. This is wild type grown on cellobiose. You don't see any induction of the cellulases. This is wild type grown on avicell. You see huge induction. And we also see that induction in the triple beta-glucosidase knockout on cellobiose. And that pattern is essentially identical as avicell. And so the proteins are identical as wild type grown on avicell. The activity is identical. And actually, the pattern of expression of genes when you expose this triple beta-glucosidase mutant to cellobiose is essentially identical to wild type exposed to avicell. So it really recapitulates the whole system. And these are just showing the enzymes. Here you can see the triple beta-glucosidase having the same pattern and by mass spec, the same protein composition as wild type grown on avicell. So the signal, we believe, is cellobiose. Cellobiose is actually at very low levels when Neurospora breaks down plant cell wall materials. And, and what we think is there's a difference in Km between the beta-glucosidase and the transporters that bring in the signal that are, or the cellobiose signal that are important for induction of cellulase activity. And so now I'm going to tell you a little bit about how we think the signal is working. And so when we grew Neurospora on plant cell wall material, we identified a number of transporters that were induced. And two of these, 801 called cellodextrin transporter 1 and CDT2 for cellodextrin transporter 2, were very highly induced. And so we knew by experiments that I'm going to tell you later that these actually are capable of transporting cellobiose, cellotriose, and cellotetrose. And our model was that perhaps these transporters were actually functioning as receptors and involved in the signal transduction to telling Neurospora it's on lignocellulose material. And this is really taken from Saccharomyces cerevisiae, where Saccharomyces senses what carbon source it's on by using these membrane-bound receptors that either can transport or function only as a receptor. And so we tested that hypothesis by looking at the CDT1 and CDT2 mutants. And as you can see in the CDT1 mutant, the response to avicell is really the same as wild type. But if you do that in a CDT2 mutant, you get no induction of the cellulases. And so this is even in the presence in CDT1 that we know transports cellobiose. So this implicates the transporter CDT2 in the signal transduction mechanisms to the cdr1 transcription regulator. So I've told you a lot about the regulatory mechanisms associated with plant cell wall degradation of Neurospora, and that's great in terms of understanding the process. But does that have any utility to biofuel production? And so one of our hopes is that if we understand the process in Neurospora, we might actually be able to transfer some of this functionality to Saccharomyces, which is the preferred, at least currently, preferred fermenting organism to make ethanol. And so I'm going to tell you a little story, finish with a story about that. So currently, ethanol is produced by simultaneous saccharification and fermentation. And what happens is lignocellulosic material, or cellulose, is added. A bunch of cellulases from trichoderma is added. Cellulose is broken down to cellobiose and glucose. But for Saccharomyces to use it, it has to be broken all the way down to glucose, which is very difficult if you think about lignocellulosic material. In addition, glucose inhibits beta-glucosidase, and cellobiose inhibits the function of cellulases. So we thought that if we added the yeast plus the cellobiose transporter plus an intracellular beta-glucosidase, that we would enable yeast to be able to use cellobiose, and higher order oligosaccharides, thereby producing more ethanol than just a yeast strain that could utilize glucose. And it would also decrease feedback inhibition into these enzymes. And so how we did that is we took those 10 transporter genes and we expressed them in Saccharomyces cerevisiae, which cannot grow on cellobiose, so it was a very easy assay. And we also included an intracellular beta-glucosidase from that we knew that was highly expressed and functional. And theoretically, this strain would be able to transport in cellobiose. Cellobiose would be cleaved to glucose, and you'd get growth on cellobiose and ethanol production. So two of these transporters, the ones that I mentioned already, CDT1 and CDT2, allowed Saccharomyces to grow on cellobiose. And this was also associated with better production of ethanol. And so how to do this experiment, we took Avicel, we added the enzymes from cellulolytic, which is a trichoderma enzyme mix. That produces cellobiose and also glucose. Now normally Saccharomyces can only use the glucose to make ethanol. And so that's this case where you have glucose utilization, ethanol production, but the cellobiose is not really used. But when we add the cellobiose transporters plus the intracellular beta-glucosidases, both glucose and cellobiose are utilized, and you get a much higher titer of ethanol. Now, one of the problems with Saccharomyces is that it prefers glucose over every other carbon source. And so a problem with utilizing plant cell wall material is you can't co-utilize xylose, which is a C5 sugar, and glucose, which is a C6 sugar, because Saccharomyces will preferentially utilize glucose. So these were done in collaboration with Jamie Kate at the EBI. And so Jamie and I collaborated with two yeast fermentation engineers at the University of Illinois, Champaign-Urbana, Human Shao and Yongsoo Jin. And they put the cellobiose transporters into a strain of Saccharomyces that can also utilize xylose. So as I said, normally when you have C6 and C5 sugars present, Saccharomyces can only utilizes the C6 sugars preferentially, and so mostly these fermentations currently are done in different tanks. So in these two papers from Human Shao and Yongsoo Lim, who are the primary authors, when they put these cellobiose transporters and beta-glucosidase into Saccharomyces, they actually got much higher ethanol production because the C5 sugars and the cellobiose could be utilized simultaneously to generate more ethanol. And so this reduces the need to degrade plant cell wall material to glucose and also prevents glucose repression of xylose utilization. And so this strain can be grown in a single tank, and you can do simultaneous fermentation. And so this strain is actually being commercialized by BP Biofuels. So cellodextran transporters allow simultaneous utilization of xylose and cellobiose. So these are the people. This initially started with a single postdoc in my lab, Xiaogang Tian, who's now a professor in China, with Will Beeson from the Kate and Marletta lab. That has then expanded. So Kate, Tian and Will initially did the transcriptional profiling of Neurospora on miscanthus. Will and Chris did the really beautiful story on the GH61s and cellobiose dehydrogenase story. Sam and James did all the transcriptional profiling on CDR1 and CDR2. Elizabeth did the beta-glucosidase story. Jianping did the CRE1 and Xylin R story, and John Galetska was the lead on the paper on the cellobiose transporters and Saccharomyces. And this has become a collaborative project with a whole bunch of people, including Chris Somerville. And these are our collaborators at the University of Illinois and also from BP, Xiaomin Yang. So thank you very much for your time. I'll be happy to take any questions.
Have you looked at all at the
Oh, thank you.
Have you looked at all at what contributes to the regulation? What conditions are necessary to express peroxidases for lignin?
Peroxidases?
For lignin deconstruction.
So Neurospora modifies the lignin, but does not degrade lignin. So I had an undergraduate who actually worked on that quite hard and looked at all the various knockouts for various oxidative enzymes in Neurospora. And it's clear that it modifies it, but it doesn't really utilize it. So the utilization of lignin is really restricted to the basidiomycetes primarily.
Okay, and so did he find any conditions that it just generally expresses it just to modify it, or is it?
Well, I think what's interesting is that in nature, for the organism to get at the cellulose and hemicellulose, it has to break those linkages with lignin. And the enzymes that actually do that are not really clear. And so we need much better assays for a lot of those enzymatic activities, and I think that perhaps some of those hypothetical proteins might actually be involved in some of those processes, or some enzymes with sort of general functions that we don't know exactly what they do.
And just one other question was, how's hemicellulose affected the expression of the cellulases, if you know at all how?
Well, Neurospora, and so it's kind of split. So many of the filamentous ascomycete fungi have compartmentalized expression of their genes. So the exposure to cellulose induces both cellulases and hemicellulases, but exposure to hemicellulose only induces hemicellulases. So that's true for Fusarium, which is kind of distant from Neurospora. So I think there's a clade that have compartmentalized, whereas Aspergillus and Trichoderma, there's some slop, so that hemicellulose induces some hemicellulases and some cellulases. So,
last question here.
You shown conservation between CDR2 between ascomycetes. Have you looked at basidiomycetes? Mechanism of regulation similar there?
No. So there are a lot of zinc binuclear cluster transcription factors in ascomycetes and basidiomycetes, and we didn't find anything in basidiomycetes that we felt comfortable that we thought would be a true ortholog. I think some transcriptional profiling of basidiomycetes might give a hint for the identification of a potential ortholog of CDR1 and CDR2.
Okay, thank you very much.